Good morning. Good morning, everybody. So welcome back to our seminars. Coffee break with TRANSFAC. Grab your coffee or tea. So I have some tea. Okay, so today we'll talk about interesting things. Let me share my screen. So we'll talk about mutations in cancer and how TRANSFAC can help with that. So basically, I will start with presenting some answering to previous questions. Basically, there was one question: how should we use TRANSFAC to analyze mutations in cancer? Because that's really a tricky question. But in meantime, you please just put your new questions in this Q&A section, and I will watch carefully what is appearing there. So, and I will answering your questions just as they are, or we'll wait maybe a little bit finishing my stuff and then answering your question. But don't hesitate, just drop your questions, and if something not clear in my explanation, just interrupt me with your questions and I will try to answer. So the goal is basically to find a really connection with you and to see what are your problems, and using TRANSFAC and basically using analyzing mutations in cancer. But any other questions also welcome. Okay, so let's start back. Let's go back a bit to our beloved motifs, which we are looking at in DNA sequences, and as you know, in TRANSFAC and as in several other databases, we accumulate motifs in the form of positional weight matrices. Sorry. So that's here you see approximately how it works. So at the end, you have such a logo which explains which nucleotides are more conserved in the binding sites for a given transcription factor. For instance, in this case, we are talking about AP1, a transcription factor which binds in genome to many places. And then first, what we do, we collect binding sites for AP1. We align them and we count how many how many cases we observe a given nucleotide in a given position. For instance, in the first position, in this case, we see always T among these 5 known sites. Maybe if we increase the set of known sites, it will be a bit more variable, but at least this gives us indication that this position has a maximum importance, I would say. Then out of these counts, we calculate frequency, which basically dividing by number of observation, and more complicated bit. So we build so-called positional weight matrices, which can be built in many different ways, and using logot scores, more classical, or this match score, which is we are using in our tools, in our match suite or in our site search tools in the platform. So we are using a bit more complicated formula. So basically, we multiply this weight, the natural weight of the nucleotide. We multiplied it by this information content, which increases additionally the importance of the conserved nucleotides. So that's how we calculate this score. And the score actually works like that. So as I said, we built such a matrix, and that's, for instance, a matrix for NF-kappa B binding sites in TRANSFAC database. So you see these weights in that matrix. And then basically, we use this matrix using this formula, which is I presented before, to search for binding sites in any sequence in genome. And this could be known regulatory sequences like promoters of genes, or known enhancers of a given gene, or maybe actually unknown ones. So for instance, you have a hypothesis that around the gene, in upstream regions, in 3' regions, maybe in introns, you can have regulatory. So we use this motif, we use these positional weight matrices to search for the binding sites. So when basically we observe this score higher than given cutoff, which are pre-calculated or computed on the fly, which I will show today a bit, then you will see you will predict this binding site. And of course, what we are going to talk today are about the mutations. So what happens if there is a certain mutation, maybe a somatic mutation? In case of cancer, we very often dealing with somatic mutations. So those changes of DNA which happens in the tumor during the evolution of the tumor. And then what we know and what people are currently using often are checking whether those mutations are hitting a particular protein, changing the amino acid of that protein, or maybe which will lead to change to the function of that protein. So in very important mutations known in cancer, like BRCA mutations and P53 mutations, enormous amount of them, which are observed in cancer, they are happening in the proteins. So they are actually changing the function of the proteins, or in some cases, in many cases, actually, they abolish a function, like, for instance, the P53 often actually destroyed by the function of the P53 is destroyed by the mutations, but not always. And then P53 doesn't work as a protector of DNA, and that leads to the development of the cancer. So, but those mutations which are happening inside the proteins, inside the genomic regions encoding proteins, they are pretty rare. Well, they are, if you look at each cancer genome, you see a lot of proteins changing, but actually very few of them are known to be associated with a particular disease or a particular cause of that cancer development, or maybe kind of used as a prognosis. There are few of which are already known, but if you look at the real cases of cancer, that luckily in many cases, if such mutations are found and they can lead to prescription of particular drugs, that often works, but in majority of cases still we don't know whether those known mutations are not observed, or simply there are other mutations in genome which are not really affecting proteins, but they're affecting the cancer growth, and they may become the driver mutations, actually mutations which are located in the regulatory regions of genes. And that's the mutations we are going to analyze using TRANSFAC. So basically, if such mutations hit a binding site of a transcription factor, basically changing one nucleotide, say in the reference genome or in the genome of that person which he has, and in that tumor, this nucleotide is changed in that position, for instance, from G to T, then obviously, since this G was very important for binding, in this case for NF-kappa B, then this will lead to a change of this predicted score, and in reality, it will lead to the drop of the affinity of that transcription factor to that binding site, which may lead to basically abolishing of that binding at all. In other cases, and we are talking about, in these cases, we're talking about actually a loss of the site loss. But pretty often, we can observe a site gain. In such a case, for instance, imagine if there was a nucleotide T in that position and there was no binding sites for NF-kappa B, but in the cancer, suddenly this T is changed to G. So it increases the affinity dramatically, and this leads to the potential of NF-kappa B to bind to that place and start regulating the gene, which should not be regulated in the normal case. So that's so-called gain in function, when the genes, which has to be silent, suddenly start to express. And then why they're expressing? Because the transcription factors which are not regulating of whatever reason. There are many reasons, actually. There might be this regulatory region is completely blocked by chromatin, or methylation is actually blocking it. But for instance, if there were no sites and a region is open, but there was no binding site for NF-kappa B, but now it suddenly appears, that it leads to the possibility that this factor would start regulating that gene, and it start increasing expression. And if this gene is, for example, the gene which leads to the growth of the tumor, then it provides the growth. That's why it's important to find those binding sites where the mutations are changing the potential binding affinity. So basically the score, which we can calculate using our algorithm. But there is another actually possibility for cancer to grow. Actually, it's maybe not necessarily this particular mutation is changing that particular binding sites of the transcription factor. So as you know, and I was talking about it many times, transcription factors, they are not actually acting alone. For instance, NF-kappa B to be active, it should have several different cofactors. And that's true for many other transcription factors. So they need cooperation. And then there might be several mutations in that region which destroy not just maybe that site is intact, but it destroys other sites, or maybe distances between sites, or some kind of looping, a possibility of DNA looping. So basically destroying a possibility for those factors to cooperate, or maybe creating such a possibility. And that's why this whole region became changed. And this again can lead to the change of regulation of the corresponding gene. So it's very important to look not just to single binding sites for transcription factors, but for regions which are occupied by many mutations. And actually what we see, and you will see it online, that very often those mutations are actually clustered. So they are not really evenly distributed over the whole genome, but you really clearly see clusters of mutations. And then you basically may assume that those mutations, they destroy or create a completely new environment for transcription factor binding style, for transcription factors to bind. And that changed dramatically the regulation of those genes. So, and we have another algorithm which is basically called composite module analyst, which is using genetic algorithms inside. And what it's doing, it's trying to given regions of DNA and given we call them yes and no regions. This could be, for instance, promoters of upregulated genes, or enhancers, or maybe regions of ChIP-seq enrichment. So any regions which we believe have a similar regulatory capabilities. But in the case of mutation, we consider regions around clusters of mutations. And we call them yes sequences. And for comparison, we take random regions of regulatory regions of other genes where we don't observe mutations in cancer in that particular tumor, and then this algorithm start comparing compositions of predicted binding sites in those regions compared to the regions which we are comparing this with the background sequences, and it finds enrichment of the clusters of the compositions of those sites. So okay, so that's a little bit of theory which we had which we are using here to analyze mutations of cancer. And if you have questions to that, just don't hesitate, just drop it in Q&A. Maybe it's just a small question I can quickly answer. Or just think about your questions and put in Q&A. Okay, today I want to actually introduce a tool which we have developed. It's called Genome Enhancer. I will introduce it very briefly, and I hope I will have more time to describe it in more details. Today, I will just demonstrate how it works for the case of mutations in cancer. And that tool is our dream of having everything automatic. That's why I have this nice slide. Basically, it's a dream of having an automatic car, which drives you to the goal, to your place where you want to go. So the same way we created actually a system, we call it Genome Enhancer, which you just drop there your data, omics data of various types, and then it works automatically until it finds, actually it analyzes those data, predict binding sites, predict clusters of binding sites. It's actually going further to see how these changes in transcription factor binding sites may lead to the change of the regulatory circuits in those cells in general. And then it goes into the algorithms which analyzing those networks and identifying so-called master-regulators. So the sensitive points in those networks, which can regulate big cascades, I would say. And then based on those master regulators, the system picked up those of them which can be used as targets for DNA, for different drugs. And then it also goes further and actually drop a list of potential drugs which can be used towards those targets. And that can be done for a set of omics data, maybe which you are doing on your cohort of patients, or maybe on cell lines, or on your experimental systems. And it also can work on individual data. So on omics data taken from one patient, it can also work and can lead to the prediction of targets, individual targets for that patient, and it will rank those medicaments which can be used in that particular cancer type from a point of view of these individual targets which are expressed and which are there to be as a master regulators of the processes which we observed here. So I see something in the chat. Okay, that's from technical support. So basically asking you to put more questions. Good. Okay. So and then this is an interface of Genome Enhancer. I will go soon online. And interesting, as I said, you can put many different omics just by drag and drop here those files as from transcriptomics, epigenomics, proteomics, mutations, and as well as metabolomics data if you have. It could be either of them, so maybe only transcriptomics or only mutations or any combination of those omics types. And actually this is a big illustration of that complicated workflow which is running under this Genome Enhancer. It's pretty straightforward. Actually, it goes from your data to the keynotes, to the master regulators, and to the prediction of the drugs. But it has a certain internal structure depending on which type of data you're putting. It makes its own decision how to proceed. Okay, so that's what I wanted to show on slides, and I will go now online. Okay, so I will go to our website, geneXplain.com. And as I said, we are talking about Genome Enhancer as a product. So you go log in. And so in our previous sessions, we were going here into the databases or directly into the platform. And we will see this platform today as well. But here you see this Genome Enhancer. And we go there. And then we log in. So you see that interface, which you've seen on the slides. And here are my projects. And today I will talk about one particular project, which is devoted to lung cancer. And I took the data from a cell line. Here you see the the name of that cell line. And as you know, basically all those cell lines, they were derived maybe sooner or later. Yeah, some days ago, some time ago, they were derived from real tumors, from real patients. Of course, if this cell line is maintained for many years, it can accumulate additional mutations. But we believe that many of those mutations which you observe in those cell lines, they actually original mutations, so mutations which happened in that tumor in that patient. And it's interesting to be able to analyze such data, because similar data you can get simply from a tumor of a given patient. In case of cell lines, of course, we have a lot of data available there is a resource called Cancer Cell Line Encyclopedia, where you can get those mutations in the cancer. You can get many other omics data about those cell lines. And what I did today, actually, I I didn't just took mutations, but I took mutations observed in that cell line together with actually transcriptomics data. So which gives me information about which genes are upregulated or downregulated in that particular cell line. And now I want the system to combine these two types of data, and you will see how they interplay. So, okay, so I just dragged and dropped these two files, and I showed before how to do that. So the system automatically recognizes the type of the data. So you see this VCF. This is a standard format for inputting mutations and genomic mutations, and it recognizes them and gives that it's actually genomics data. And here an Excel file containing already pre-processed data from that cell line, giving the basically a fold change to comparing this expression of those genes in that cell line, comparing to actually a normal tissue. So as a normal tissue, it was taken, we took it from that encyclopedia. We took actually as a normal tissue, the actual lung tissue. It's actually very specific comparison, of course. But anyway, if you have patient data and where you can really compare expression of a gene in the tumor compared to the expression of those genes in the neighbor tissue of that tumor, or in the tissue of that patient, which is where this tumor is sitting. That would be a better comparison. But of course, in case of cell lines, you have to compare to some other data, and then here we compare it with a normal tissue, expression of those genes in the normal tissue. All right, so we go next, and this is just how a system works. You take your data, you drop it here in this box, and it shows you this in which table which columns this table have. So, for instance, in the case of of that table, which I dropped here, we have this transcriptomics data with fold change here, log fold change, and you see these upregulated genes and downregulated genes, and you have here p-value and adjusted p-value. So standard transcriptomics data. And we have also put here these genomics data, which are actually topic of the discussion today. And if you click on that type of data, you will see them in genome browser, which you see here. So on a chromosome, you can also put here more information, but for that we'll look at that later. Okay, but what we'll do next, actually we go next. And now we form our run. So for that system, we actually take the mutation data, and we take the log fold change from that table of transcriptomics data, so that system knows which column from the table to use to compare to take upregulated genes on downregulated genes. By the way, if you don't have this pre-processed data and you have your raw data, for instance, as counts, then you can also use it here in next maybe seminars. I will show it in more details. So the system will automatically compute this logfile change by you. If you have it already computed, you can use it directly as I showed here. So, and then you click next, and this is a form which you should fill, which is a very simple form. You select here the disease you are looking at. So in this case, we have like lung cancer, lung neoplasm. Actually, it was non-small lung cell lung cancer, non-small cell lung cancer. Sorry, non-small cell lung cancer, and that's the disease we are studying here. And now you have to select that combination of data which you prepared to run, and in this case it's just that set which we just prepared. If you would you would have a normal tissue and cancer tissue, and you could compare things, then you can make several conditions and then system would start comparing. But here the comparison was already done. Mutations were detected in the tumor, and the fold change was already calculated for the tumor. So we have just one condition, so we actually don't need any other conditions. And we also activate a special module of that system which will compute also a special report which tells us about the known mutations in proteins, known and actionable mutations in proteins. We call it molecular tumor board report. Okay, so and then you simply start. So it will proceed with analysis. It takes maybe several hours, 2-3 hours, and pretty long because it has inside many complicated algorithms, also including this genetic algorithm, which takes time to optimize the information. So we will not wait for this long. So of course I did already analyze this data. I will stop that run. So and I will look for the report. So here the full report, it's called full story. It has a lot of information in it, and we will spend, I think, special time of analyzing this information in that type of report. It actually analyzed your data and create a summary, which already telling you the most important master regulators which were found by the system, and those transcription factors which were either changed by mutations or in the regulated regions of upregulating or downregulated genes, or which are actually in clusters in these combinations are observed around those mutations, around those clusters of mutations. Okay, but what we will talk today is actually the part of that report which concern the mutation, the analysis of mutation itself. So you see it's actually generating such a table. We can open this table in full. Actually, it gives you a list of genes where the mutations were observed in clusters. So you see, those genes which have multiple mutations, and they are located. So you see, what you see here are the blue blocks are corresponding to exons of the genes. These arcs, they corresponding to the introns, and you see here these 3' and 5' regions, and the red bars indicates the positions of the mutations. And actually, you see some mutations are located in introns, and some are in exons. And actually, for instance, here it's definitely in some coding region of each exon. But sometimes even they are in exons, they may be in a non-coding part of the exons. So we really separate, the system separates those mutations which are located in exons and in coding regions of exons, and those which are located in regulatory regions. Good. Oh, okay, I have one question. So the question is very interesting. By which authority is Genome Enhancer certified for application to patient data in hospitals? That's a very important question. Actually, that's very this tool is experimental, so it's provided as it is, and it's not yet registered by medical authorities to be used in hospitals. So researchers may use it for their own purposes, and of course, it's at the end, if they find some hypothesis using this tool, this could be this must be verified and must be actually used by the doctors on their own responsibility. It's like using Google. You find some information in Google, and you can use it or cannot use it in your decisions, but at the end it's responsibility of the doctor to make its own decision. So it's a supporting system to get some additional information. But that's important question, of course, and that's tells us that that's a very important area and very sensitive area because actually you understand right. So when you're dealing with such important thing as tumors and cancer patients, that everything has to be balanced, and regulatory authorities has to be involved in that. All right, so okay, and we go on further. So you see those mutations which are observed not only in exons but also in introns and 3' and 5' regions of that gene, of those genes. And then what happens next is actually it starts calculating whether those mutations which are located in not in the coding regions but in the other regions of genes, they may affect whether they can they can kill or destroy the binding sites for transcription factors, or they can create the binding sites. So here you see the column which tells us about the losses, basically when the mutations are destroying binding sites. And here you see gains. So when mutations actually creating new binding sites. And here you see this is an indicator or index, or we can call it name, for that transcription factor family. So which is described by positional weight matrix. So that's, I didn't tell you, but probably you know already that it has such a complex name. So V stands for the vertebrates. So in TRANSFAC we have matrices for various phyla, so for plants, for Drosophila a bit, and but most of the stuff there is about the motifs in vertebrate genomes, and mainly in human, mouse, and rat, but also sometimes in other mammals genome. Then we have a name of the family of transcription factor family, sometimes of an individual species of transcription factor, but in most of the cases a family, a group of factors, which have a very similar DNA binding domain. And then there is a certain code here which just tell the number, consecutive number, or it has sometimes a bit more information, like Q5 means this matrix was constructed by our team, and it was the construction of that matrix was based on the binding sites with quality better than quality 5, which means more or less all of the known transcription factor binding sites. And in fact, this number means this matrix was taken from the literature as it is, and which means for ELK, for instance, there are several matrices, and matrix number 1, 2, 3, and so forth. So sometimes up to 20 and 30 different matrices for one transcription factor family, taken from different resources, from different databases. So actually, that's how these matrices are named. And then here you see how many cases among those mutations, among those sites actually, which were predicted in the regulatory regions of those genes, we observed the loss of that site or gain of that site due to mutation. So we, for instance, for flea, we observed only 179 cases when the mutation was actually creating a binding site for flea. but 3,000 something mutations when these binding sites were destroyed. And that's called losses. And we can estimate the p-value, or the probability of such an event happening by random chance. And if it's like 10 in minus 17 or 10 in minus 9, it means it's very unlikely that such thing happens by random chance. So what does it mean? Actually, it means that you have a lot of mutations in various places in genome, but for some reason, many of those mutations, they tend to delete the binding sites for Fli. Why this could be, why this could happen? It's like a miracle, you know, all the mutations in many places in genome, and suddenly there is some power which prohibit those sites to be there. And I believe that we believe that this power is actually the selection which happens in tumor. So, you know, tumor starting from the very beginning and to the end, to the very growth, it goes through a very rapid selection, a rapid evolution. So this genome, tumor genome, is under very strong selection pressure, and actually, which means that for some reason, for instance, this Fli transcription factor is very active, right? So it maybe gets active in those tumor, and it starts binding to many places and dysregulate a lot of genes. This may be a protective selection, which stabilizes the genome, or it can be a driver mutation, a driver selection, which leads to, you know, if Fli is so important, for instance, it can activate genes which facilitate the growth of the tumor. Then these Fli binding sites will be incorporated, will be mutated, so will be created, by the way, by this selection, and then we will see this effect in the statistics, which actually tells to us two things. So first is which we will know which binding sites to look for and in which genes this happens. But also what is important for us is actually which transcription factors are involved. For instance, this list gives us a list of transcription factors which get active. Because if they are not active, why should be a selection in that genome towards either inhibition of the binding site for those factor or creation of the binding sites for this factor? So if selection is going in either of direction, then it means this factor is actually active in genome, is in fact is a sort of an indicator about activity of those transcription factors. And as you know, activity of transcription factor is not necessarily just expression of that protein, of that transcription factor, but also, which is very important, that this transcription factor has to be activated. So it has to get a specific phosphorylation or other type of modification which makes this transcription factor active, and it would start binding to the target genes. What is done then to activate those transcription factors, we'll talk about a bit later. But let me check, what is your question? Can we predict the effect of these gene mutations on drug binding to targets? Right, that's a very interesting question. Well, in this particular example, no, we're actually talking about you know, how mutations are changing transcription factor binding sites, right? But what you are talking about the drug binding to a target, right? So actually, drugs usually binding to the targets, which are proteins, right? So if you have mutations in the protein which changes this binding of the drug to a protein, then of course they are affecting. But here we are talking about mutations not in the protein, but in the promoter of that protein, promoter of the gene which encode that protein, right? But in fact, it's more complicated, because which we observed very often. For instance, I studied resistance of tumor cells towards a drug. I think it was what was that? So a particular drug which has a very important targets, right? And what I see actually a mutation in that target itself, so which can change the binding of the drug to that protein. But what I also see mutations in the promoter of the gene which encode that target. And what happens often in the cell lines or in tumors, that, for instance, the tumor gets a mutation which in the protein which abolish the drug binding. So basically you get drug is not getting effective, right? But in the same time, you can the genes could start expressing more of that target, which would compensate this abolishment of drug binding. So the drug binding to the protein gets less, but the amount of the protein get more. So, and this could compensate one another. So the mutations in the promoters of that gene can in fact also affect the drug target interaction in a sense. But okay, thank you for that question. Let me go further and close this window. Okay, so we see this table, and actually this table is even bigger. Well, it has 35 entries, which lists all the motifs, all the transcription factor, binding site, position weight matrices, which we found either statistically significantly lost or gained. Actually, very interesting what you can observe, of course, in the example like this one, very extreme, when it's definitely, we are talking about in practically in all cases of destroying of the sites for this factor. Maybe it's very important activator. And very few really, when these sites were, they are lost, but very few when they are gained. But in many cases, in some cases, you can have both p-values are very significant. For instance, E2F. And is no, E2F is a transcription factor which regulates cell cycle, and it's very sensitive point. And in some genes, this site has to be increased. In some genes, they have to be decreased. And then both directions are under the selective pressure, and you can get a lot of sites which are creating a lot of mutations which are creating that sites, and a lot of them which are destroying those sites. So, but this both indicate that E2F is getting important in that tumor. All right, so that's how this statistics is calculated, and it gives us a list of transcription factors and their position weight matrices, which get activated in that cancer, in that tumor. We are talking about cell line. But actually, there is a second step, which, as I said, I told to you that it try now to take the whole region around the clusters of binding sites, and it start analyzing the compositions of binding sites, not just individual binding sites like for E2F like this, but with which cofactors, with which other binding sites this whole system work together. So, and for that we run this composite module analyst, so-called genomic algorithm, which try to find the composition of binding sites in the regions around clusters of mutations, and comparing them with those regions which doesn't contain those clusters of mutations. And on this histogram, which is well, you see clear distribution, the discrimination between red bars and blue bars. It's not the best, so sometimes and very often you can see much better discrimination, but in this case it's significant. So, but you see a lot of noise there still, but nevertheless it's AUC is 0.77, so it's a kind of higher than the random, and then you see those. So what does it say actually to you? It gives a certain score, actually, whether cluster of mutations can have those binding sites around those clusters. And if you see many cases, many genes which have those clusters, they have a very high score of that combinatorial score. So which gives you basically an idea that those genes are really regulated by those clusters of sites. Okay, so we can look a bit more in depth. For that, we click on that table, and this table shows to you, we can sort by the score. Actually, this is a combinatory score I was talking about. And what does it show to you? Actually, for each gene, and here we have actually all genes, 8,300 genes, which have upregulated in that system. It's actually in those cell lines, it's quite a lot of genes. Not all of them are significantly upregulated, but these are basically all genes which have a fold change higher than 0. So basically more than so positive log fold change. All right, and and then for each gene we have here a promoter of that gene. All right, so it's around 1,000 nucleotides upstream from TSS, and then each of this little arrow is a predicted binding site for that cluster which I was talking about. So this cluster of positionally weighted matrices. So we have here how many 12 matrices, 13, and here we see then the binding sites for those different matrices, and then you see how many of those matrices gave the binding sites in that particular promoter, and it also look for the clustering of those binding sites. So this dark area, this shady area, demonstrates you actually that in this place we observe the cluster of those binding sites for different transcription factors. And this score is telling you how many of those matrices found their sites in that promoter, plus how good they are in terms of clustering. And you see sometimes these are more distributed. Here you see more sites, but they're actually more distributed along the promoter. Sometimes they are more clustered than in this case. So, and then you get information about which sites are really located in the promoter of that gene. Right. Okay. So we can, I think at that moment, we can also look at a bit more details, even more details. I will go now into the platform view. So now, so far, we're just looking more or less information from that report and looking at these ready tables. But if you go now to the platform view, we in fact can go directly in that folder where this system, this Genome Enhancer as a pipeline was running and created so many outputs. So actually it created this report, which we are now looking at. And it's also created a lot of other files, intermediate files. You see them all here, which were run by many different programs inside this pipeline to analyze the data. And I just showed you very small part of the analysis, the part which is concerned with transcription factor binding sites, how it is affected by the mutations, and so forth. So let me show one example of that. For instance, we can go to complete predicted size. So actually, if we go here in the genome browser, we can drag and drop those predicted sites here. Okay, so and then what we do, we can put here also annotation. We put here the gene annotation, so we can maybe do it like this. Also, we would prepare here a little bit. So let's go to tracks, and let's take out of these names of the mutations because they don't give too much information. So we see them just little dots. And if we now jump into predicted sites, So we can go different chromosomes, right? And now what we do, we will go to one particular gene, which I prepared for you. Let me have a look. Actually, there was one interesting gene, which is a ribosomal gene, which is upregulated in that cell line. And I was, by looking at this data, I was interested in that gene. So I will put here this okay, let me see my, copy it correctly, ensemble. So it should be ensemble ID of that gene. All right, so let me, okay, so I'm wondering why my view is empty. That's something to point, you know, look, this is a 37, so it's a wrong build. So you have to be careful with looking at the right build. So let's open the correct build. So for that, we basically do this like this. So these are genes from the build, from the last build of the genome, so 38. And now we actually will put here the data again. So we can put here our predicted sites, and we will put here the mutations which we brought, right? So, and for the mutations, we go to the track and remove these names. They already removed. Okay, very good. And now we will go to this gene. And then we will see what is going on here. All right. All right, so let me see. So we can, you see, we have this, let me see, that's not the gene which I wanted to have, but just to demonstrate you, actually this gene was also important, but maybe it's not, it's less interesting because here we see that mutations are actually a bit far from that promoter. So what this system is doing, so in case of mutations, when they are actually located inside the gene, like in this case, so for instance, these mutations are located in the intron of that gene, right? So then and if this gene is upregulated in our system, then it will analyze that region around those cluster of mutations. But if the gene which is upregulated, in fact, is not, there is no single mutation which is in that gene, it will just take a promoter of that gene and analyze how this promoter of that gene is look like. So that probably was another good example. Let me find another one. I prepared a couple of examples actually for you. So that's a gene for so-called TMED3, and that's very interesting gene, in fact. And if we have time or you have questions, we can talk about that gene. It's known to be involved in different cancers, not in lung cancer. So that's, by the way, the cell line is about lung cancer. Sorry, I didn't tell it in advance. But what you see here is those mutations which are located in the basically in promoter or even more extending 5' region of that gene. They are located nearby. So, and then the system analyzed binding sites which are located in that whole region and found a cluster of those binding sites near this mutation. So we can have a look. For instance, this mutation is located between these two TIF2 binding sites, but we see here the clusters of other sites which are not affected by mutation. But as our system analyzed, and it found out that these factors are very important to work together with TIF2. So it means that this mutation may influence the work of the whole system. So that's the hypothesis which can be generated here. So in this case, for instance, it's just one mutation, but in other cases, you may observe clusters of mutations in those regions. Okay, so that's just a little of grasp of information you can get from such analysis. You can explore your tables and you see which genes are upregulated. You can go here into the genome browser and see in which cases those mutations are located in the whole region, or in which cases they may be affected by the binding sites, or binding sites are affected by those mutations. So they are co-located with the binding sites. So you can zoom up to the nucleotide level and see in case of the mutation is actually hitting the binding sites, you will see it also. So you can go also by yourself through those information flow, and see all those cases. So what I want to show today at the end is what we do. Okay, we found here transcription factors, they are binding sites, they may affect the expression of the genes. We see the correlation between the mutations, clusters of sites, and the upregulated or downregulated genes. So we create hypotheses which transcription factors can be important for regulating the genes in this tumor. So what? So what is next? Transcription factors are nice to publish, but do we have anything to do with them? And as I said, there is a big part of Genome Enhancer which then works further. So it starts thinking, okay, these transcription factors are got active. So what is the process which makes this transcription factor active? And it creates, we call it starts the upstream analysis, and it creates such maps of the networks. We can show it a little bit, more in details. It creates such networks, which, here at the bottom of that network, you see these genes which are upregulated in that cell line. You see the transcription factors, which are obviously working in the regulated regions of those genes affected by the mutations. And then you see the cascade of signal transduction, which may lead to activation of those transcription factors. In that cascades, you see, we call them master-regulators. For instance, you see here cyclin B, you see BRCA1, you see other transcription, Aurora B, for instance, EGFR. So very well-known genes which are involved and, which are markers of prognosis, which are actually related to the therapeutic targets. But in this particular case, in this particular cell line, we found them very important as potentially regulating those activity of those transcription factors. And in turn, they are regulating expression of those genes, including the genes which encode those proteins by themselves. So that's the network which is created by the system. And based on that network, we predict which of the master regulators can be actually potential targets for the therapy. And we go further, and we go further and predict which drugs can target those master regulators. And you see here well-known drugs like erlotinib, gefitinib, which are also FDA approved for lung cancer, and especially for this type of lung cancer. And in this case, we also look at this paclitaxel, a drug which is very often used and also FDA approved. But for this cell line, the system predicts that this drug has very high rank. In addition to quite well-known, tyrosine inhibitors, tyrosine kinase inhibitors, like this paclitaxel is in number 3. And we were wondering why is that. And when we look at into the data coming from cell cycle, from cell line, cancer cell line encyclopedia, where they which is correlated to, or related to another resource, which were in NCBI, people were analyzing which drugs are affecting various cell lines. And when we took this cell line, you see this is a cell line we're analyzing, and we check which of the drugs from those assays have maximum effect on those cell lines, basically killing them with even very low concentration. At the top, you see this Paclita cell. So it's experimental fact that Paclita cell is really, that this cell line, this particular cell line, is very sensitive to that drug, and we see that obviously in our calculations when we analyze expression of genes plus mutations in the genome observed in that cell line through that knowledge gathering from our different databases and reconstruction of networks. We predicted that the targets, first of all, the targets for Paclita cell can be predicted here as a master regulators, and many of them, and in total this score of this targets altogether for that drug is very high, and that's why in this rank this drug was ranked high. Okay, so that's a bit deviated from initial story about transcription factors and binding sites, but I think it's important just to tell you to which direction we are going. So not just studying cell lines and publishing paper, we want to actually be useful here and predict to which extent we can predict how these tumors and cell lines are sensitive to various drugs and which targets are actually active in the particular tumor, and then that may help to find new drugs and new therapies. All right, so I made my speech. It was pretty long, 1 hour. I'm sorry for that. So I didn't give you a chance for your questions, but we have time. If you still have time to sit with me, and maybe you also, by the way, if you are watching this video in YouTube, you people in future, you who are watching it in YouTube, please don't forget to give to click on like, and also maybe you can subscribe to our channel. That helps. But anyway, I will wait for your questions a bit. Don't hesitate. So I will go to the website. Yeah, the question, is this service free? So we have here several products in our system. So what I was talking today was Genome Enhancer, which is not free. So you have to subscribe it, and that you can find here on the website how to do that. So click to get subscription and to buy a license. So we have various models for the Genome Enhancer. You can buy the license for the whole year, or you can buy it for a shorter time. So that's all possible. As for the free products, we have the GeneXplain platform. We have a possibility to get a free account. So it's your lifelong account. You just create it and you get it. You get there is a very easy form. You get this. You fill it and you get immediately the password. So you link where to go. And this account has quite small space. It's only 15 MB, where you can put your data and analyze your data. It of course doesn't have commercial databases like TRANSFAC and TransPass connected to this account. So you can later or immediately you can add those subscription, but you can also work with your data. You do a lot of pre-processing, and I was talking about this a product before. You can also read about that in our website. You can do a lot on this free account, so don't hesitate, just go and try it. So otherwise you're You can also send to us any inquiries, price requests, and so forth. Okay. All right, so you have here, so let me go through our products. You see our list of products. It looks a bit complicated, maybe, but actually we have an easy navigation, which is we have the big table, which talks almost about all our products. So which is like we call it TRANSFAC packages. So we have what I was talking about today is actually the full package, which includes Genome Enhancer and all the subscription to all other databases like TRANSFAC, TransPass, and human PSD, which is the information about drugs and targets and these kind of things. TRANSFAC is about transcription factors, their binding sites. TransPass about signal transduction and networks, and human PSD is about drugs and their targets. But you have smaller packages which just include these, TRANSFAC only, or TRANSFAC with platform together, and so on. So you can see, you can read about the advantages of each of the package. And yeah, I would recommend to take the full package. All right. Any questions? By the way, we have also, it's a bit related to the topic today. We have other products which are not in that table. For instance, we have a products from chemoinformatics. One is called PAS, which is actually a very interesting product. What you can do if you are working in chemistry, in the drug development. So that's a product which I would say pretty many companies are buying. It's you have your library of compounds. You are interested, your own library or library from other providers. You load it into this program, and it creates very quickly, so in milliseconds, it creates a prediction of for each compound. It predicts effects, like whether it's anti-tumor or effect on a particular target, and so on. So almost 5,000 different effects in general it can predict, and then it selects those drug-like molecules which can be important for your activity which you want to predict. So that's PAS. So please have a look at this. Okay, so there people are thanking me. Thank you very much also for listening. So that's one product which might be of interest. And another product is which was also not listed in the table is Brenda. That's actually a database on enzymes, and it's a huge database on all enzymes. This Brenda is produced in Braunschweig University, and now it's moving also here locally. And actually, that's a very important resource for those who are interested in actually metabolism, and not only human, mouse, rat, or it also goes very much beyond that. It talks about bacteria, a lot of different bacteria, a lot of different enzymes in those bacteria, what they are doing. So very interesting database. You can be providing it on our servers. We provide additional services. It's also it can be also you can get it also for free from the university itself. But if you are doing your research and you want to use our service and our servers, secure servers, you can subscribe for that database also by us. Okay, so I think I don't see many further questions. Anyway, so you can send more questions to us just offline, and we will consider them next time. And actually, I will also prepare some interesting questions which we are constantly getting from customer support. Yeah, so okay. And right, so you can send it and we'll try to answer it next time. Thank you very much, and thank you for your thank yous. And okay, we'll see you next time. Goodbye.